we look through the circulatory system, I'll mention things about cardiac output. If something reduces cardiac output, that's lowering the blood flow through the system, and it can inhibit or reduce delivery of materials. Also, when you think about the circulatory system, what's its job? I mean, like I said, all this system is doing is like a mechanical irrigation system. You're pumping fluid from one spot to another, but why are you doing that? Of course, the main role of the circulatory system is it's a distribution system, it's a transport system. It moves things from one place to another. What does it move? Well, the list would be huge, all right? I mean, the hormones we just talked about, how do they get from place to place? Circulatory system. What else do you transport with your circulatory system? All of your nutrients. Carbohydrates, glucose, amino acids, that sort of thing, all the nutrients. What else? Waste material. Nitrogen waste has to be moved from where it's produced to the kidneys to get rid of it. So it's definitely, a transportation system. However, things like hormones, nutrients, waste, that's all important to transport. It's true. There's usually just a couple of things we emphasize when you're looking at the efficiency of the circulatory system, and that's the two respiratory gases, in particular oxygen. So when we're talking about the circulatory system, we'll often just mainly discuss the transport of oxygen and carbon dioxide. Why those two? Why don't we talk about nutrients much? It's not that the other things aren't important. It turns out transporting respiratory gas, and particularly oxygen, is really, really difficult. This junk is hard to move in sufficient quantities to supply an animal. So if you have a distribution system that can get enough oxygen to your tissues, everything else is going to be fine. It doesn't matter. Easy to transport nutrients, waste, that sort of thing. But the oxygen is difficult. So if you have a transport system that can move oxygen, everything else is going to be a piece of cake. So that's usually why they emphasize, oxygen and CO2 when we're looking at how well the circulatory system works. All right, let me start looking at some pieces. And I'll get back to cardiac output, because anytime we talk about, how's this going to affect the heart, you kind of have to pop back to that cardiac output equation. Take this up here real quick. So what we'll start off with, we just kind of look at some parts of the heart. It's kind of what we did in lab, so it's sort of review stuff. But we'll throw a little bit of function as we go. All right, so, here's our heart. All this is the mediastinum. I mentioned it lab, it's on your lab terms. It's one of those terms I'll probably never ask in lab, but this column of tissue between the lungs, sternum will be out here, vertebral column back there. That whole thing is the mediastinum. That's where you find the heart. And if we kind of peel the other junk out of the way, we'll just look at the heart itself. So the heart is located in the mediastinum, kind of in the center of the chest. It deflects a little bit to the left. But what we're going to do is imagine if you cleared those lungs out of the way, we'll just go through and look at the layers real quick. Kind of look at these in lab, but I'll add a little function to it here. So if you're starting from the outside and we're moving in, the first layers associated with the heart are the serous membranes. These are the pericardial membranes. They've enlarged it up here. So what you have is you have a heart. Here it is. That looks more like a prostate, but it'll do. And then there's a double membrane around the heart, like all the serous membranes. You have a layer that is physically attached to the outer surface of the heart. That's either the epicardium or the visceral pericardium. So here it is right here. Then it folds back on itself. And there's an outer layer that's loosely fitting. That's the parietal pericardium. That setup is the same for all the serous membranes. Visceral pericardium or epicardium, you can use it either one, it's the same structure. This is basically a layer of epithelium and basal membrane along, attached to the outside of the heart. It's fairly delicate, like other serous membranes. Then you've got this space in between the two membranes. That's your pericardial cavity. That's filled with the serous fluid, so you look at a fresh heart, that heart can slide around that bag, very little friction. Again, that's serous fluid and reducing friction. That's a function of all the serous membranes. But then what's a little odd with the parietal pericardium, it's made up of 2 layers. I'll draw my ugly version, and then we have the pretty one on the board there. All right, this outer layer, kind of right here, it's got an inner serous layer, kind of that yellow bit there, my black bit. That's just, again, epithelia that secretes the serous fluid, you know, like any other serous membrane. But then you have this outer, right here, this fairly thick fibrous layer. Down here is kind of the blue layer here. These 2 terms are on your lab sheets, but again, you can kind of tell they're there in lab, but we don't look at it in close enough detail to really separate them. So there's a fibrous layer, and inside of that, there is a serous layer, and this collectively is the parietal pericardium. This is odd. Most serous membranes, you wouldn't have that serous layer, or that fibrous layer. You'd have the visceral layer, and you'd have the serous layer of the parietal layer of the pericardium. But why do you have that fibrous layer on the outside? You know, if you look, some of those cats, you could pick up the cat by the parietal pericardium. It's that tough. It's basically the same stuff as, you know, the fascia on the muscles. You know, it's a dense, you know, connective tissue. A lot of collagen fibers, very strong. Why is that out there? That has absolutely no function in lubricating the heart. And it's not, you know, you got bones around the heart, so it's not protecting it from external, you know, punctures or anything. So what's that there for? It's kind of interesting because, again, you don't see this on any other serous membrane. That parietal pericardium, it's protective in a way. The way it's protective is a little odd. Give you an example. When we talk about the uterus, I think I gave you a curve something like this. We did a leg tension curve with smooth muscle at one point, didn't we? Okay. So I think I did something like this here. Why did the uterus skeletal muscle? Because skeletal muscle has a leg tension curve. Would look something like this. That is, if it's really short, it doesn't generate much tension. If you stretch the muscle, so it's really long, which is relative scale. If you overstretch it, it doesn't generate much tension, right? So that's a typical length tension curve for skeletal muscle. Smooth muscle, you know, I said it did something like this. That's why it's in hollow organs like the uterus. You can stretch smooth muscle like crazy. When you stimulate it, it still contracts with the normal amount of force. Now what about the heart? All right, the heart is striated muscle. It's got the same arrangement as skeletal muscle. That is, it has sarcomeres with actin and myosin filaments. You know, it's striated when you look at it under the microscope. So if you start stretching out cardiac muscle, will that affect the amount of tension it develops? Yes. It's just like skeletal muscle as far as its length tension if you look at the, you know, isolated muscle. Which is weird because most of your other hollow internal organs are smooth muscle. The heart is not, it's striated. Now what's interesting though, if we took an intact heart and tried to do the same thing, do a length tension curve. Keep in mind, length tension curve in the heart, length is really circumference. Let me slide down here real quick. I just have this. If you look at the way the fibers are arranged, of course the heart muscle fibers aren't linear. They're wrapped around, you know, the chambers, right? So when you think of length in the heart, length of muscle fibers, it's really circumference, right? Another way of thinking of that is this hollow space in there is where the blood goes, right? If I push more blood in there, what happens? I fill it up more, I stretch it more. Is that increasing the length of the fibers? Yeah, the circumference. So when we talk about length for the intact heart, it's not really length. Think of this circumference, or functionally what's more important, filling. So when we think of linear length in a skeletal muscle, in the heart, it's basically filling. If you push more fluid in it, it stretches out more. If there's less fluid in it, they're going to overlap more. So if we took an intact heart, and we did sort of something similar, length-tension relationship, although it's really, you know, circumference or filling tension relationship. we did the same thing, we'd get a curve that looks something like this. Only half of it there. Now, if you think about it, this is critical for the heart. If you could fill the heart up enough and get it like skeletal muscle over here, guess what's going to happen? It won't generate any force. If you overstretch it or overfilled it, all of a sudden, boom, no cardiac output because no stroke volume, it couldn't contract anymore. You keel over dead. You can or definitely don't want to overstretch the heart. So if you look at an intact heart, this is the length tension curve. If you fill it up or stretch it a bit more, you get more tension. If you fill it up a little bit more, you get more tension. Fill it up more tension. More tension. More tension. That's it. You can't go any further. Why not? That's what this bag is for. That parietal pericardium's fibrous layer is not stretchy. It's collagen fibers. It's relatively, I won't say rigid. It won't stretch. It's got a lot of tensile strength, but if you pull on it, it doesn't give like an elastic structure. So what happens is you can fill the heart up a bit more, it'll contract with more strength. Fill it up more, contracts with more strength. More strength. So cardiac muscle has a length tension curve, which is critical. I mean, we'll get to it in a bit. But as you increase the filling, it's when it contracts, it's going to contract with more force up to this point right here. The reason it stops at this point is it bumps up against that fibrous layer, and that's it. This is really important because it prevents the heart from going on this downstream end. It physically can't do it because that parietal pericardium prevents it. In fact, what's kind of cool, let me stick another picture up here. This one, you won't want to read it up here, but this is posted to Blackboard. It's a little article. You can read this if you like. I'm not going to read it to you, but this is an article out of, I think it's an old cardiology journal. But it's kind of interesting. It's a short little abstract talking about the effects of the parietal pericardium. What they've done here, they took greyhounds. You know greyhounds, right? Those racing dogs. They run around the track chasing some great rabbit or something. Right, they exercise a lot. There's some money in it. So this guy did some experiments looking at the effects of the pericardium on cardiac output and oxygen delivery to the tissue. Again, when you're looking at aerobic athletes, whether they're dogs or humans, generally the more oxygen you get to your tissues, the better your aerobic performance. Typically, in most mammals, diffusion is great. We are diffusion limited. We're perfusion limited. What limits our aerobic capacity is how fast our heart can get blood and oxygen to the tissues. So, oxygen delivery, blood delivery to the tissues, or cardiac output is going to affect aerobic performance. So what they did with these greyhounds, they took a couple of groups. In one group, they opened up the chest, they removed the parietal pericardium, chucked it, closed them back up, let them heal. They took another group, and they did what's called a sham operation in these studies. They basically cracked the chest, did nothing, put it back together. That's just to show, cutting into the sternum and things like that could have an impact. It's sort of the control. Anyway, they let these dogs recover, and then they raced them with monitors monitoring blood oxygen and cardiac output and that sort of thing. And what they found is the dogs that had their parietal pericardium removed performed better because that parietal pericardium limits stroke volume. Remember, cardiac output is determined by heart rate and stroke volume. Well, the stroke volume has a limit because of the parietal pericardium. If you remove it, the heart could actually fill more, and when it contracts, it ejected more blood. So a dog running without parietal pericardium, it could actually increase a little bit more its stroke volume. So that thing actually limits your cardiac output a bit. Now, this is not a long-term study. They didn't tell you what happened to the dogs 5 years ago. Probably not a good thing long-term, but at least in the immediate term, that thing, we know it limits stroke volume a bit. And that's, evolutionarily, that's its purpose. It prevents you from going down here. You stop the load. I'll also tell you, this is kind of the difference between like an athlete and a non-athlete. Like, if you look at me, I'm kind of a couch potato. My heart rate is, let's just say it's 82 beats per minute. Now, if you got an athlete, this could, a competitive athlete, like a, high school cross-country runner that's, places in events, that sort of thing. We'll just call them athlete. Their resting heart rate might be 50 beats per minute. Couch potato, athlete. Heart rates are quite different. So who's pumping more blood just at rest, sitting at the couch there? No one. We are pumping the same amount. We are pumping about the same amount because at rest, everyone's oxygen demand is basically similar. So both of us have to pump 5 liters per minute. I have a heart rate at 82, they've got a heart rate of 50. So what has to be different if we're pumping the same amount of blood? Stroke volume. Remember, cardiac output is heart rate times stroke volume. What you'll see with athletes, they will typically have a much slower resting heart rate. Why? Because their heart, it expands more, and when it contracts, it ejects more. So they don't need to pump as many times, they don't have to beat as frequently to pump the same amount of blood. That's a change that occurs with training. Like heart rate, anyone can get their heart rate up. I mean, if I go up a flight of stairs, boom, 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 it'll go real fast. Anyway, that's just nervous system. So it's really easy to up heart rate. Changing your stroke volume, now while everyone can increase stroke volume some, we'll talk about that briefly, but that's what changes with athletic training. If you do a lot of cross-country running, things like that, one of the things that happens, the heart gets literally bigger and the parietal pericardium gets a little bigger. That allows you to increase your stroke volume. That's kind of the difference between athletes and non-athletes. They can all have a heart rate, but the athletes have worked on that heart muscle itself. It's bigger, it's got a bigger stroke volume. You can read that if you want, I thought it was interesting. So, cardiac muscle has the same sort of leg tension relationship as skeletal muscle. It's just terminated by that top fibrous parietal pericardium. So it limits how much an intact heart can expand. All right, let's see. So that is the pericardium. So you got the outer, still called the parietal pericardium, just know it has 2 layers. Outermost, coming from this direction, the outermost is a fibrous layer of the parietal pericardium. Then there's a thin serous layer of the parietal pericardium, and then that folds back, and then you have the serous layer of the visceral pericardium, or epicardium. Those are the outer layers. Now let's move into the middle layer. Most of the heart's volume out of this part on this diagram is myocardium. These are cardiac muscle cells. Want to take a minute to review some of the characteristics of the cardiac muscle. You looked at it a little bit in Anatomy 1, probably real quick. You looked at the muscle slides. That's cardiac, that's smooth, that's skeletal, gold. But let's look at some differences, because there are some important characteristics of cardiac muscle cells. In fact, I'm going to put a little table up here. Let's just compare them to skeletal muscle. You've done skeletal muscle. I realize you've probably forgotten most of it since last semester, but it'll come back to you pretty quick. All right. One of the important similarities I've already mentioned, these are both striated. That means they both have sarcomeres. The contractile mechanism is basically the same. You got the Z lines, you got the actin and myosin filaments overlapping, you get cross-bridge formation, you get troponin that moves tropomine, all that is the same. They are striated, same sort of organization of protein fibers. That part is the same. And of course, they're both muscles, and they both generate tension. What's different? You remember some differences? Give me a few.
Intercalated discs.
Okay, that's probably the big one from a histology perspective when you look at those slides, if you have a good one. Intercalated discs are found in cardiac muscle. They are not found in skeletal muscle. Now, what is the intercalated disc? Basically, if you have a cell here, and I'll throw another one here in red. Here's another one, another cardiac muscle cell. Cell 1, cell 2. You have a cell-to-cell junction. Right here, you got one cell attached to, an adjacent cell. So this connection point, of course, when the cardiac muscle cells contract, you don't want them pulling apart, because that would suck. The heart would kind of fall apart. It wouldn't pump anything. It would be terrible. So this cell-to-cell junction, that is where you find the intercalated disc. And the reason you can spot it, at least if the slide is stained properly, there's all kinds of proteins at that cell-to-cell junction. Some of them, not going to be important to us, but we'll mention them. There are, if you remember desmosomes maybe, there are belt desmosomes. Those are just big proteins that kind of knit the two membranes together. It gives them mechanical strength. So when you generate some tension, it will tear apart. So there are desmosomes in there. They're just tight, sort of cell adhesion type of connections. But then functionally, the one that's important for us, also in the intercalated discs, there are some proteins that form gap junctions. All right, the gap junctions are going to be functionally kind of critical for the heart. If you remember, gap junctions is a protein that kind of rolls into a tube and it connects the cytoplasm of one cell to the cytoplasm of the next. Gap junctions are usually described as being used for cell-to-cell communication. Every cardiac muscle cell is connected with these gap junctions. So in effect, what you have is one giant cell. All the cytoplasm is connected. That's really functionally important for the heart because, as we'll see, just like skeletal muscle, what do you have to do to make this skeletal muscle contract? What has to happen? You send a nerve impulse to it, right? There's a neuromuscular junction. You depolarize that skeletal muscle cell, right? And that depolarization is what activates it, all right? If you take one skeletal muscle cell and you took a couple little electrodes and you stimulated one skeletal muscle cell, so you got a skeletal muscle cell here, and you got another skeletal muscle cell here, and you got another skeletal muscle cell here. If I stimulate this one with a couple of electrodes or with a neuromuscular junction, if I stimulate electrically, it'll depolarize and contract, right? What will this one do? Nothing. What will this one do? Nothing. They are not electrically connected, which is what you want in a skeletal muscle. Do you want all these fibers contracting at the same time? Boom! No, it'd be really hard to feed yourself with a fork because you jab yourself. I know, you don't want it. You want to, graded contractions in the whole muscle. You can generate impulses in a couple cells for a little tension, more cells, that recruitment thing, that's the motor unit recruitment. So you can change the amount of tension. The heart, on the other hand, what would happen if a few cells depolarized and contracted and the others didn't? And then another few cells depolarized and contracted. The heart would just sit there and quiver. It wouldn't function. In order for the heart to effectively squeeze on the blood, every cell pretty much has to contract at the same time. The gap junctions allow this. Unlike skeletal muscles, they're all electrically connected. So if I had a bunch of cardiac muscle cells, I'm just going to borrow this one, gap junction here, gap junction here, gap junction here. So now all the cytoplasm is sort of connected. If I stimulate this cell, that action potential, the movement of sodium in and potassium out, that's going to flow from this cell to this cell to this cell. They'll all depolarize because they're all electrically connected. And then they'll all contract. This is what makes a pacemaker work. You know what a pacemaker is? If someone's heart rhythm is off, they'll get a pacemaker. In its simplest form, a pacemaker is like a little battery and a couple of electrodes with a timing circuit. The electrodes are stuck in one cell, right there. And then when that thing zaps this cell, that's the reason they work. The impulse travels to every cell, so they all contract at the same frequency. If you put a pacemaker in a skeletal muscle, it would just make those few cells go, it wouldn't be communicated. So these gap junctions relay the information. The heart is actually working almost like one giant cell. I'll call it a functional syncytium, I think is the word they might use in the textbook. But it's working like one giant cell. It's a unit because it's all connected with these gap junctions. Skeletal muscle cells don't have them. What else is different? Oh, it's a few histological things as well. Linear, cardiac muscle cells usually branch a little more. Kind of a little bit more branching. Oh, remember the nuclei? Skeletal muscle cells are usually referred to as multinucleated. There's dozens of nuclei in a single cell. Cardiac muscle cells are typically mononucleated, or occasionally they have two nuclei. But they're not multinucleated like a skeletal muscle. They're usually a little bit smaller. They're more branching. What else is different? Cardiac muscle, skeletal muscle. One thing I will mention, metabolic. Do your skeletal muscles work anaerobically, that is using glycolysis and fermentation? The answer is yes. Anytime you're maximally exercising, your big muscle groups, like your skeletal muscles in your legs, they will burn the oxygen way faster than your circulatory system can deliver it. So if you're sprinting as fast as you can, and you jab a syringe into your leg muscles, and you pull out, it hurt a lot, you pull out a sample of fluid, you check the ice, it's going to be low. Skeletal muscle work anaerobically. It's no big deal. They can use glycolysis and fermentation to get 2 ATPs for every glucose molecule available. It works fine. Of course, the downside is that as you build up that lactic acid, and eventually you get muscle fatigue, right? That oxygen debt you build up. Cardiac muscle cell cannot work anaerobically, period. It can't make ATP by glycolysis and fermentation or any anaerobic means. Which means if cardiac muscle cells run out of oxygen, what happens to them? They die in like 2 minutes. That's what a heart attack is. So these are strictly aerobic metabolism only. Your cardiac muscle cells cannot use fermentation and just glycolysis. They got to use Krebs cycle, electron transport chain to work. Which seems a bit like a bummer, because if your cardiac muscle could work like skeletal muscle, you could shut off the oxygen supply for a while, and it could work anaerobically and still make ATP, there would not be any heart attacks. Wouldn't. Because the only reason you have a heart attack is it's usually a temporary blockage of blood and oxygen. Those cells die in a few minutes. Once those cells are dead, they're gone. If you could work anaerobically, no big deal. Go anaerobic, make ATP using glycolysis and fermentation. So why not, any anaerobic metabolism? Well, think about it, it kind of makes sense. When your skeletal muscles go anaerobic, using glycolysis and fermentation, eventually what happens? They fatigue, they develop an oxygen debt, build up of lactic acid, other metabolic end products. Then they have to stop contracting. They lose the ability to generate tension. Then there's a period of time we have to pay off that oxygen debt while they're sitting there doing nothing. If your heart worked anaerobically, when's it going to pay off its oxygen debt? It can't. If it stops, the rest of your body dies and it wouldn't be a good system. That's evolutionarily why. Your heart can't work anaerobically because it could never stop to pay off an oxygen debt, at least not in mammal. So just aerobic metabolism only. What else? Difference between cardiac. There's a couple others I'll mention, but there's one big one. So you got the gap junctions in intercalated discs. That connects all the cardiac muscle cells together. What's also cool with cardiac muscle, if I cut off this bicep, slap it on the table, what's it going to do? It's just going to sit there, right? But if you were quick and cut out some mammal's heart and set it on the table, what's it going to do? Just like in horror movies, it still beats. You can sever the heart, pull it out of the chest, and that sucker will still beat. In fact, I got another class that work on some frogs once in a while. We do this frog experiment, we take the heart and we apply epinephrine, acetylcholine, and a few other chemicals. But you can take the heart out of a frog, cut it out, drop it in a jar of saline, set it on the counter, and 3 hours later, that sucker's still going in a jar of saline. That's creepy. Skeletal muscle will never do that. In order for a skeletal muscle to contract, what must it receive? Some kind of outside signal. I have a term for it. Skeletal muscle is referred to as neurogenic. Just means a skeletal muscle normally has to get input from a motor neuron. If there is no input from a motor neuron, skeletal muscle won't do anything. That's why you get paralyzed. You sever the motor neurons going to skeletal muscles, that's it. They're now useless. They won't work. Cardiac muscle is a little different. The term is myogenic. It's myogenic. What does myogenic mean? It means the signal that initiates contraction does not come from outside of the heart. It comes from the cardiac muscle cells themselves. This is kind of weird. So you could, in fact, take the heart, sever every nerve to it, and it would still function relatively normally. Now, we'll get to it eventually. There are some nerves going to the heart. They're just to modify its rhythm. They are not there to initiate it. So you could, in fact, cut every nerve going to your heart, and you'd still function pretty much okay. Because the signal that makes the heart contract doesn't come from outside of the heart, as we use the term myogenic. Well, that causes a bit of a problem. If you think of any of the skeletal, cardiac muscle, there's a cell. If you remember, this is sort of review from last semester, but if you forgot it, that's all right. You remember when you were maybe looking at skeletal muscle, they have a resting membrane potential. Skeletal muscles, neurons, sort of the same thing. It's somewhere in the neighborhood of -70 millivolts, right? That is the resting membrane potential. Most cells have this. It's not unique to muscles and nerves. Most cells, you stick an electrode inside and outside of the cell, you'll measure the electrical potential of somewhere around -70 millivolts inside relative to outside. You know, the magnitude varies a bit by cell. -70 heart cells are actually closer to -90, I think, but it's about the same process. You remember why how does the inside of the cell get negative? You remember that? The ions that are used? You probably looked at 2 ions. For example, sodium ions tend to be at a really high concentration outside of cells. If you measure the sodium ion concentration in the cytoplasm cell, it's really low. It's a fraction of what it is out here. Any other ion that you might have looked at was potassium. All right, potassium. Typically very low outside of the cells. Inside the cytoplasm, you know, it's often more than 10-fold higher. It's a lot of potassium in the cytoplasm. All right, suffice it to say, you know, you move these ions around. There's a difference in the distribution of ions inside and outside, so typically the inside is going to be negative. It's got fewer positive charges or more negative charges. However you look at it will work for that. So you have this resting membrane potential. Now, all of the skeletal, all the muscles, skeletal muscle, cardiac muscle, they're the same. Remember, these cells have a threshold. You remember what threshold is, maybe with the neurons? It's a set point. If you recall, and this doesn't matter if it's an axon, skeletal muscle, or cardiac muscle cell, they kind of all have the same thing. One of the things they have in their membrane is a voltage-gated sodium channel. You vaguely remember those from action potentials, is probably where you've done. A sodium channel is just, it's a protein in the membrane. It has a hole in it that's just the right size to allow sodium to move across the membrane, but it like has a lid on it. And it's normally closed, so sodium can't move anywhere. But if you stimulate this little protein, it'll pop open. And so it's just like a hole with a lid on it. It's normally closed, but if it opens, it's going to allow sodium to rush in. That's what depolarizes a cell at the beginning of your action potential, right? Well, these are called voltage-gated sodium channels because what they're sensitive to is membrane potential. You know, proteins have a specific three-dimensional structure, right? That's all this is. They have a particular conformation. If you affect the outside environment in a proper way, you change its conformation, which in this case opens it. This thing is sensitive to the membrane potential across the membrane right there. Now, normally, this membrane potential is -70 millivolts. At that charge, that gate is closed. But if you can depolarize the cell a little bit, you know, make it become more positive inside, there's probably a threshold, obviously TH for threshold, of about, let's call it -50 millivolts. So if I can get this membrane potential 20 millivolts more positive, this thing's going to pop open, sodium floods in, then the potassium channels open, potassium floods out. That's your action potential, right? Just kind of ringing a bell. I'm not going to even worry about the details, I know what's different. But that setup is the same in an axon, in a skeletal muscle cell, or in a cardiac muscle cell. They all have these voltage-gated sodium potassium channels. And if you get this thing to threshold, they'll pop open, you get an action potential. In a muscle, if you get an action potential across the membrane, that's what causes calcium to be released by the troponin, and you get cross-bridge formation and contraction, right? Depolarize the muscle, it contracts. That's true of skeletal muscle, that's true of cardiac muscle. If the muscle depolarizes, it will contract. When it repolarizes, it relaxes. The difference is, what makes a skeletal muscle reach threshold? That's that motor neuron forming a synapse that opens some sodium gates temporarily at the synapse, that depolarizes at the threshold, then the muscle contracts. That kind of makes sense? It's just that neuromuscular junction. Cardiac muscle cells don't have that. So somehow, here's the tricky part, we have to get from -70 to -50 without the input from a motor neuron forming a synapse with this muscle. So that's what's different. They both have more or less similar type of threshold, similar type of resting membrane potential. They both generate an action potential the same way, and when they depolarize, they both contract. They repolarize, they both relax. All that's the same. The difference is, how do you get the threshold? How do you get the cell a little more positive without a motor neuron, like the skeletal muscles do? Again, skeletal muscle is neurogenic because it's a motor neuron that brings in the threshold, then they depolarize and contract. With the cardiac muscle cells, they don't have that. So what do they have instead? It's another type of protein. I'm going to draw it over here. These are, think of them as like little pinholes. They call these things sodium leak channels. It's descriptive. These are like little pinholes. They're always open, but what do? They allow sodium to slowly leak in, because like I said, it's sort of like a pinhole. It slowly leaks in. Well, as sodium slowly leaks in, what's going to happen to this resting membrane potential? It's more positive, more positive, more positive, so you hit threshold. That's all you need. In fact, if you were kind of recording it with an oscilloscope or a data logger or something, it might look something like this. Now you've got, we can put time down here, and membrane potential here. So we got -70, we'll call that our resting membrane potential. -50, that's our threshold. Resting membrane potential. And somewhere up here would be depolarized, say +30, something like that. All right, normally in a skeletal muscle, you'll stay at the resting membrane potential until that motor neuron sends a signal. That brings the threshold, you get an active potential. But in the heart, there's no nerves going to it, but you got that sodium leak channel. As sodium leaks in, it gets more positive, slowly gets more positive, slowly gets more positive. Because sodium's leaking in, bringing in positive charges, right? And as soon as you reach threshold, then these big honking, voltage-gated sodium channels open, just like they would on skeletal muscle, and you get a massive depolarization, then they close, then the potassium gates open, and you're back in. But then the sodium leak channels are still there. So you slowly get more positive, slowly get more positive, slowly get more positive, slowly get more positive. Oh, you hit threshold again. But then you get the idea, you get more positive, more positive. It'll just happen. That's how you get your heart rate. You can just take a heart cell, put it in a petri dish, and watch it. It'll just contract by itself on some set frequency because of these sodium leak channels. This one allows them to be myogenic. So you don't need input from the outside. These things will still contract. An isolated heart and a jar of saline will still keep beating. Now again, we'll look at it later. There are still nerves going to the heart, but they don't initiate the heartbeat. They modify it to make it go faster or slower. They don't initiate it. That's all comes right from the heart. Down here a minute. All right, this is a skeletal muscle cell. We looked at last semester. Just want to mention a couple other differences. They're not terribly critical for us, but a little bit. Do you remember? So this is one cell. They removed the plasma membrane here. So what you're seeing are internal, cell components, organelles. All this blue junk here, that is smooth endoplasmic reticulum. In the skeletal muscle, they rename it, but it's still smooth endoplasmic reticulum. You remember sarcoplasmic reticulum in skeletal muscles? What was its main function in skeletal muscle? Calcium storage, right? This is where there's massive amounts of calcium ion stored. When the cell depolarizes, basically calcium gates open, calcium floods the sarcomeres. You can see here, binder's component, you get contraction. Kind of ring a bell? All right, the mechanism is very similar in cardiac muscle. It's not sarcomeres. It's like this. They're still dependent on calcium. But what's kind of weird in cardiac muscle, this sarcoplasmic reticulum, while it's still present in cardiac muscle, it's much, much less. So it has less, I'm just going to call it SR because I don't want to write out sarcoplasmic reticulum too many times. It has less of it. Now, it still contains calcium, and again, when the cardiac muscle cell depolarizes, that SR does release calcium, binds to troponin, you get contraction. But there's not enough. So what that means is, for a skeletal muscle, all the calcium it needs to bind to all the troponins to get maximum contraction comes from within the cell. In cardiac muscle cells, there's not enough. There's not enough calcium stored in there. So when the muscle is depolarized, yes, calcium leaves the SR, but there's not enough to saturate the troponin. The contraction will be weaker. So guess where the remaining calcium for a heart muscle cell comes from? That's why parathyroid hormone is so important. Comes from the blood. It's extracellular. So unlike skeletal muscle cells, cardiac muscle cells are dependent on extracellular calcium. Every time that cardiac muscle cell depolarizes, some calcium has to diffuse from the outside, from the blood, into the muscle, into the sarcomere to bind to troponin. So 20% or so of the calcium needed for maximal contraction in a cardiac muscle cell is extracellular or blood calcium. Again, that's why when I talk about parathyroid hormone, that's why it's important. Skeletal muscle doesn't care at all what the extracellular calcium is. It isn't dependent on it. But cardiac muscle, if your blood calcium levels are too low, there's not enough diffusing into your heart, it will contract with less force. It'll be weaker. It's less sarcoplasmic reticulum. All right, kind of related. You might have looked at it a little bit last semester. I'm not sure. It's not a big deal. But you remember what the T tubules were for in skeletal muscles? T tubules are transverse tubules. What was their main function? Look at these little yellow things here. What are they for in skeletal muscle contraction? You remember?
Transporting the
Well, in skeletal muscle, they are for, here's your skeletal muscle. Remember, skeletal muscles are huge. I mean, if you measure them in inches, they're very long. They're a fraction of a millimeter in thickness. I mean, you can see them with your naked eye. They're big. You know, a magnifying glass and a needle, you can separate out individual skeletal muscle cells. They're huge. And the potential problem here with these huge cells, the action potential, here's a motor neuron. You know, if you fire this motor neuron, you get an action potential traveling across the membrane, just like you would on an axon. The problem is, remember, those action potentials, that electrical difference is right next to the membrane. If you move far away, there's no electrical change. It's very localized. So what about the inside of this huge cell here? Is that action potential there going to affect the cell in the middle? No, it doesn't travel. That's what the T-tube is for. All they are, they're invaginations of the plasma membrane. So they're just membrane. And so when the action potential reaches that T-tubule, it goes on down. It's a way to conduct the action potentials deep into the muscle cells so you signal all of the sarcoplasmic reticulum to release that calcium at the same time. Now, cardiac muscle has the same mechanism. You know, the action potential, well, it doesn't start with a motor neuron. It's generated by the cell itself, but it still sweeps across the surface of the cell. It does have T-tubules for the same purpose. It conducts the impulse deep into the cells, but also the T-tubules, if you kind of compare them, I'm exaggerating a bit here, they're huge in diameter compared to ones on skeletal muscle. That's one of the main reasons why, the reason you mentioned. Not only do the T-tubules in cardiac muscle cell have to conduct the action potential deep into the muscle, but yeah, this is also a site where some of that extracellular calcium can diffuse in. It's making a bigger surface area, so extracellular calcium has an easier way to get into the cardiac muscle cells at every heartbeat. Those are the main differences. So very similar mechanically to skeletal muscle, but it has some metabolic differences, and some of the details are a little different for generating its own action potential and such. But again, the actual mechanism, we look at muscle fiber contraction last semester, that's the same in cardiac and skeletal. It's got the sarcomeres, troponin, tropomyosin, all of that is the same. All right, so that is the myocardium. Again, the myocardium, the muscle, that's what actually contracts, generates the force, takes up the bulk of the heart volume. And then the innermost layer, just mentioned it briefly, that's the endocardium. The endocardium just covers all the internal surfaces, lines the chambers, covers the valves. It's thin. It's just like an epithelial layer at the base of the membrane. Endocardium main function is to produce a smooth surface, so that there's not a lot of friction blood flow. That's pretty much all it does. That's endocardium. So those are the layers. And now, we've got a little time, so let's look at some pieces and parts. All right. I'm going to start off with this diagram. I won't sit real long. Well, these diagrams look more realistic. Because the heart is kind of three-dimensional, it's really hard to figure out what's going on, because things are running behind each other. But let me start off with this one. All right. So we look at the heart in lab. It is a pulse pump. You know, it's like a Dixie cup made of muscle. It fills up with blood, then it contracts, it squirts the blood out of a hole, and it has a rest phase, so it's cyclical. There are some names for the cycle, not a big deal. When the muscle is contracting and squeezing on the blood, we use the term systole. It's just contraction. So anytime you're talking about the contraction in the heart muscle, you'll see it written as systole. Systole simply means the muscles are contracting, they're generating force, that sort of thing. So systole, just remember that equals contraction. That's really all you need to know. And then, of course, if the heart's not contracting, it's resting. Well, I have a name for that, too. When the heart's doing nothing, It's just sitting there like a Dixie cup under the faucet. It is in diastole. Diastole is rest. It's not contracting. It's doing nothing. So diastole, the heart is doing nothing. But keep in mind, when the heart's doing nothing, there is something going on. What is it? That's when it's filling with blood. So when you think diastole, rest, remember that equals filling. So that diastolic or the diastolic period, that's not nothing happening. That's actually when blood is filling up the ventricle, so when it contracts, it's got something to move. So there's something going on. All right, so contraction, that's when it's squeezing out the blood, pumping it out. Diastole is when it's not, the heart's not doing anything, but it is filling passively with blood. Now, when we looked at the heart in lab, remember you got a left and a right ventricle, that's what they're showing you here, and a left and right atria, right? All right. You actually have two pumps. The heart is not one pump, it's two. If you were really good with the razor blade, you could go from the apex here, cut through the intraventricular septum, cut through the intraatrial septum, and you have two pumps. There's an atrium and ventricle on the right side, there's an atrium and ventricle on the left. They are both pumps. They're too confusing, so to start with, I'm getting rid of one. I'm only looking at one side of the heart. Let's look at the right side. The right side of the heart. Now, like I said, that diagram's nice, it's pretty, but it's hard to figure out what's going on. So here's my version of the right side of the heart. Beautiful. There we go. All right, I got two squares or two rectangles. This top one here, this is the right atrium, and this bigger box is the right ventricle. That is one of the pumps. So by itself, it will pump blood. You only need one side of the heart and you'll move blood. So this is a pump. So we need some inlet plumbing and some outlet plumbing. If we're looking at the right side of the heart, what brings blood to the right side of the heart? That would be? Which ones? Blood to the right side. I think I'm half deaf because I keep hearing things. Vena cavas, say vena cavas. Sorry, I just can't hear. It's a combination. I'll blame it on the mask, not my old age hearing. All right, so there's blood vessels bringing blood to the heart. Now, by definition, the blood vessels bringing blood to the heart are veins. And these specifically on the right side of the heart, it's superior and vena cava. I'm kind of drawing as 1, 2, because I'm too lazy to make it look real pretty. So those are the vena cavas. They bring blood in. All right. Now, as we're looking at blood flows and movement of blood in general, we're going to have to look at pressure. You already told me that. You gave me 120/80 as a normal blood pressure. All right, I got two complaints. This is what I would write on the test, and my physics teachers would always get pissed at me. What is that? 120 what? Pounds? mmHg. I just want to mention something real briefly. We look at a lot of pressures with the circulatory and actually the respiratory system as well. The units you typically use in medicine and physiology are millimeters of mercury. This is not an SI system. I think that's Pascal's. We don't use it. We use millimeters of mercury. You don't know why we use millimeters of mercury? Because it's kind of an archaic unit, but we use it. It's used medically and in physiology still. Millimeters of mercury. It goes back to some of the first experiments. It's very easy to measure pressure with something called a mercury manometer. They don't let us play with them much anymore. I had a couple of my office, but they took on my mercury because it's really hazardous now, and they don't want to. Kind of makes you numb. It explains a lot of things. But all they are, real quick, squirrel here. Imagine you take a glass tube. Little glass extension out here. You take this glass tube and you fill it with mercury. Mercury is a liquid metal. It's very dense, but it's liquid at room temperatures.
Very accurate.
Yeah, they're incredibly accurate. And all you do with this thing, so all this is mercury, and you would literally take a ruler, stick it here with your little graduations on it. Whatever you want to measure the pressure on, you take a little tube, catheter, what have you, from here, and you stick it into whatever. Blood vessel, heart, a blood pressure cup. As the pressure goes in here, it's going to push the mercury down, and it will rise up here somewhere. It'll drop down here somewhere. You just measure that height difference. That's millimeters of mercury. all the blood pressure devices used to be that. There was some clinics in town, not until recently, they had this big, those sphygmomanometers, when you take blood pressure, put that cuff on your arm, and you just inflate it, and you read the pressure off a gauge. Those gauges, if they're uncalibrated, you don't even know it, they're inaccurate, and you gotta check them all the time. Mercury manometers, there's nothing to go wrong, they're spot on. One of the clinics right down the road here, I think they just closed down within the last year or two, they had this big wood box, it was a huge tank of mercury, it was a mercury manometer. So you just hook this up to your blood pressure cuff, and you get exact readings. It's millimeters of mercury. That's how we measure blood pressure. So what it means is, this 120, that means that's enough pressure to raise a column of mercury 120 millimeters high. I am sloppy, I leave out the units. You shouldn't, like me, I'm not going to mark you wrong if you get in like physics class. So no, you need units. It's millimeters of mercury, both of these. But it just means that's how high you raise a column of mercury. So it was just an easy device for measuring pressure. Until they realized if you let everyone play with this mercury, they tend to go nuts, and it's really hazardous, and they close schools now, and the kids bring it. We used to play with it in our hands. I mean, it was cool. But that's how they get millimeters of mercury in these old manometers. The other thing I want to mention is, what is this? There's two numbers here. Is the pressure 120 or is it 80? Or do you average them? Why two numbers?
Because 120 is the contraction, it's the blood going
Right, they call it
in and
systolic
then-
over diastolic pressure. So if you were actually monitoring someone's blood pressure, it would actually look something like this. You're not measuring all of the blood pressure. All you measure clinically, you measure the peak and you measure the trough, the pressure lowest and highest. It's every point in between. It's not as practical to measure that. So yeah, that 120 is when the heart is ejecting blood into blood vessels, pressure's at its highest. 80 is when the heart's not ejecting blood, the pressure drops to its lowest. We'll get back to this more in a second. All right, so pressure. We say your blood pressure is 120 over 80. What if I told you your blood pressure was 2? Are you worried?
Yeah.
Your blood pressure right now is 2 or less. See, what you got to keep in mind, clinically, we use the blood pressure in, what's this artery right here? Brachial artery. That's it for comparison. That's when we use. It's easy to get to. Your blood pressure is different anywhere you go. And think about it. If you have a tube, it doesn't matter if it's a piece of PVC pipe, copper, or blood vessel. If the pressure here is, let's say, 100 millimeters of mercury, and the pressure here is 100 millimeters of mercury, what direction is the blood going? To the left or to the right? What makes sense? Pressure's 100 here, 100 here. Why would it move? In fact, it wouldn't move, would it?
No.
If the pressure is the same, there's no flow here. The only way blood is going to move, my left to my right, the pressure has to be lower here. Agree?
Yeah,
So the pressure is the same, it's not moving. So what does that mean? If you look at your circulatory system, artery comes out, blood goes around here, goes here, comes back to the heart. If you move that pressure monitoring device anywhere in the circulatory system, will the pressure be the same? No. In fact, here it would be higher. Here, lower, then lower. Doesn't it have to get lower through the entire circulatory system? If it didn't, blood would move. It would move backwards. In order for the fluid to be flowing through a tube, there has to be a pressure gradient. So in fact, your blood pressure drops constantly. If you move that blood pressure come around further away from the heart, well, it will be small, but there's a drop. Every time you moved it, away, drop, lower, 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 lower. It's small in the arteries, but it would still be noticeable. So this pressure is just, by convention, we measure here on everybody. But if you moved it, your blood pressure is the whole range, pretty much. 120 over 80 is nearly 0 with the vena cava. In fact, I like this question on our exam sometimes, where is the pressure lowest in your circulatory system? Looking at my lovely diagram here, we know blood flows from the heart, right, out the arteries, around, 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 and it's going to end up back in the ventricle. So where's the blood pressure the lowest?
Lower leg.
Well, if it were lower here and higher here, wouldn't it go back down this way? I mean, if I had high pressure here, lower pressure here, blood would go from the veins back down the leg, wouldn't it?
Yeah.
And we know it doesn't do that. So where's it got to be lowest? Where's it ending up? In the ventricle, right? The blood flows into the heart, so it's going to be lower in the heart. Where is the blood pressure the highest? That's a trick question. Blood pressure is highest in the heart, and blood pressure is lowest in the heart. Does that make sense? It's highest in the heart when it's ejecting the blood in systole. It's lowest in the heart when it's in diastole. The blood's flowing in there. And it will change through the entire circulatory system. So just kind of keep that in mind. All right, sorry, back to my heart. So blood's going to flow from the vein. It's at very low pressure. Now let's say we're looking at the heart while it is in passage. So the heart is doing nothing. It's just sitting there like a Dixie cup under the faucet. The vena cava is kind of the faucet. So the blood will move right into the atria. And of course, the atria have a hollow spot. It will fill up. But there's a big hole between the atria and the ventricle. That's what we kept sticking our fingers through in the lab. It's called the atrioventricular orifice. So the blood's going to pour right in here. So it flows from the vena cavas into the right atria, into the right ventricle, and you will fill up the right ventricle to 80% of its capacity in diastole. So the heart is doing absolutely nothing. And the blood's just flowing into the atria, filling it up, flowing into the ventricles, and it fills the ventricles up to about 80% of its capacity completely passively. So this, again, it's like holding a Dixie cup under the faucet. The heart isn't doing anything. The blood's just flowing into it, you know, from the vena cavas, because there's a little more pressure here, not a lot, than there is in here. So it fills it up. Now, eventually, the heart is going to exit diastole, and it's going to begin to contract. Now, I know I told you earlier, all these cells are connected with gap junctions, so they all do contract at the same frequency. But, answer me this, does the axial potential move through all those gap junctions instantaneously? No. It's diffusion. It takes time, right? So in fact, while it's true the heart, all the cells contract at the same frequency, it takes time for the impulse to move from one area to the next. Typically, the impulse starts in the atria. So what you would see if you were watching a heart while it contracted, you'd see these atria contract first. You know, they'll lead the ventricles by about 1600ths of a second. So of course, when the fibers wrap around the atria contract, what they do is they make the inside of the atria smaller. They're squeezing on the blood. And that's going to take that last bit of blood and squeeze it down into the ventricles. Now that, because the timing is a little off, you may see systole and diastole referring to individual chambers. I could refer initially to this as atrial systole. The atria are in systole. Initially, the ventricles are still in diastole, so they contract first. So the ventricle fills passively to 80% of its capacity. That last 20%, they're topped off by the atria. And, you know, you've looked at the atria and the ventricles in the lab, right? How big are the atria, really? How strong are they? They're pathetic. They look like little baggies stuck on top of the heart, right? They're not much. In fact, right now, if your atria quit working, you know what would happen? Probably nothing. You might not even notice it. Now, you wouldn't win any marathons, because you're, you know, as we'll see, you lose about 20% of your max cardiac output. But you would be fine. The atria aren't that important, in quotes. Now, if your ventricles quit, you keel over dead immediately. The ventricles, you know, you saw in the lab, they're massive, huge amount of muscle. They do most of the work. The atria don't do that much. You know, like I said, if they quit pumping right now, it wouldn't have your resting metabolism could remain the same. Like I said, your top aerobic output might be a little lower, but you'd be all right. What do the atria do? They've got really two jobs. This is one of them. The main function of the atria is to top off the ventricles. That last 20%. That's what they do. So 20% of the ventricular filling is active. That's the atria kind of squishing that last bit of blood down in there. The only other thing the atria do is they act as a temporary holding tank. If the ventricles are in systole and not receiving blood, the atria can kind of fill up for a moment to hold that blood. But they're really top-off pumps. That's their function. If they quit working, I think it was George Bush, President, the senior, the first one, he had basically a situation where his atria quit working. I think he was exercising in the Rose Garden at the time. And the symptoms, he said, he was a little short of breath. You know, so it won't kill you. The atria, they're important, you know, for efficiency, but they're doing minimal amount of pumping. But that's one of their big jobs. So the atria fills up that ventricle, tops it off completely. So after atrial systole, the ventricle is now completely full at 100%. And shortly after atrial systole, it's like, it's about a tenth of a second, more or less, the 1600th of a second. After the atria contract, then the ventricles will begin to contract. Now the ventricles, they are huge. They're massive. You know, that cow heart, they're like an inch thick. So they will start squeezing on the blood, and that pressure is going to shoot up really fast, really high. But here's the problem. You got this big hunk and hole between the atria and the ventricle. Blood is going to follow a path of least resistance. So if the blood's flowing from the atria to the ventricle, it's doing that because initially the pressure is higher in the atria than the ventricle. But when these ventricles start to contract, that pressure is going to go really, really high. This blood is going to immediately try and go back from the ventricle into the atria. Of course, you can't allow that because if it goes from the atria to the ventricles, back into the atria, back to the ventricles, back, it's not circulating, it's just oscillating. You will keel over dead. So we looked at these in the lab. There are a series of one-way valves in the circulatory system, 2 of them on the right side of the heart. Their sole function is to prevent the backflow of blood. What do you call a valve between the right atria and the right ventricle? It's called the right AV valve, tricuspid valve, yeah. It's got a little flap here, a little flap here. And again, I think I mentioned lab, these valves are completely passive. They are nothing but a bit of connective tissue covered in epithelium. No nerves, no muscles, really nothing. And like I said, I like to use these doors as an analogy. This is just like a valve. Well, sort of, except bigger. Right there, but, imagine air is blowing out of this room. If air is blowing out of this room, what's this door going to do? It'll keep it open, right? What happens as soon as the air turns and blows into this room? They grab that door, slam. If you have like a loose screen door in your porch, what happens in the summer when the wind shifts direction? These valves are just like, that door, sort of. You can think of them as being hinged here, and they'll open this way. So anytime the blood is going sort of from the atria to the ventricle, it's just going to keep these cusps, these doors, pushed open. But as soon as blood tries to go this way, it basically is going to grab that cusp and it slams them shut. Now, like I mentioned in the lecture, this is the heart sound you hear. Blood flowing through the heart, it's in streamlines, it's laminar flow, doesn't make a lot of noise. You can't hear it with plain old ear. But when those valves shut, they vibrate the tissue. So that, lub-dub or thump-thump, that's the first sound. It's that AV valve slamming shut. Now the ventricle will continue to contract, squeeze on this blood. It can't get out here. Of course, there's this second opening into the ventricle. That one on the right side is connected to an artery called pulmonary trunk. Oops. That's the wrong color for the pulmonary trunk. So the ventricle will contract, squeeze on the blood, and of course it will eject it out the pulmonary trunk. But eventually, 35 hundredths of a second, that ventricle is going to now start to relax. What happens to the pressure in the ventricle when it relaxes? It goes to approximately 0 millimeters of mercury. What is the pressure? Is the pressure in your arteries ever 0 if you're alive? No. Remember that example I gave you earlier? Now this is the aorta. I'll get to the pulmonary trunk in a minute, but it'll have a similar view. You got your systolic pressure here for the aorta, that's 120. For this one, it's 80, right? All right. Here's a different example. Here's a spigot coming out of the wall here, out of your house or something there, out in the backyard. Your hose spigot. Well, let's say you attach a hose to this thing. Here's your hose. And there's a valve here. All right, now when you turn on your hose valve, water's coming out under pressure. I don't know what's your house pressure, 30 psi or something, it's pretty good. There's pressure in these lines, so the water is flowing out of this hose, right? And there's pressure in here, water's flowing. As soon as I shut this off, what happens to the flow through this hose? Drops to 0. What do you think the pressure would be? 0. So when blood's being ejected by the heart, pressure's high, that makes sense. But how come when the heart stops pumping, it's just like closing this valve, why doesn't the pressure, why does it look like this? 120, 0, 120, 0 in the aorta. Why don't it go to, when you close all those valves and the heart's not ejecting blood, why did it drop to 0? I mean, driving would really suck because you'd pass out like 70 times a minute, so it'd make it tough. But why doesn't it drop to 0? Well, I can't really hear.
It's okay, go ahead.
You can yell louder.
I was gonna say, well, because it's gonna retain pressure so the blood goes up.
Well, it is going to retain some pressure. Here's why. If I replace your arteries with PVC pipe or copper pipe, it would go 120, 0, 120, 0. It would. Because as soon as you shut off the blood flow, pressure drops to 0. But if you feel like your radial artery here, can you feel it? Yeah. How come? If I replace that with a piece of, copper pipe, could you feel it? Like if you go in your bathroom and hold on to the plumbing, turn the water on, turn the water off, can you feel it at all? I mean, you might feel the temperature change, but it's not expanding. Our blood vessels are not like PVC pipe and copper pipe. You might have looked last semester, you remember your, elastic regular tissue slide you had in histology? That's an artery. It's the only place you find elastic regular tissue. But if you look at an artery, in the walls, particularly the medium-sized arteries, there's elastic tissue in there. They're not rigid. So when the blood pressure is really high, when the heart's in systole, if you watch the artery, it's going boom. It's stretching. So using my faucet analogy here, I could do the same thing, except I'm going to put a little pipe here, and I'm going to attach a water balloon. Now, when I open this spigot, will you agree water will go down this way, but some of the pressure will be used to fill up that balloon, right? Now, if I close the valve, what's going to happen? Will the water stop flowing here immediately?
No.
Oh, why not? You've stored some of the power in this, some of the pressure in this elastic balloon, right? It will recoil, keep forcing fluid through, and it'll keep a pressure and a flow in there for a while until it's deflated, right? That's your artery. Arteries are like a rubber elastic balloon. When the heart's in systole, they're inflating like this. Then the heart's in diastole, the walls recoil, and that keeps the force going. It actually smooths out these pressure differences so you don't get quite as high and quite as low. So the reason it does drop to 0 is not really the heart, it's those arteries. In fact, one of the things that happens to us as we get old, your elastic tissue is less elastic. In your skin, in your lungs, in your blood vessels. They used to refer to this as hardening the arteries or atherosclerosis, in part, they call it still sometimes hardening the arteries. If your arteries, as you get older, lose their elastic characteristics, they're turning into more like PVC or copper pipe, which means they don't store the energy as well. So just like copper pipe, guess what happens? Your diastolic pressure starts to drop too low. That's going to affect average pressure, so you know what you do in response? You take this whole curve and you move the sucker up. So you have to shift systolic pressure up to keep diastolic pressure from plummeting too low, so your whole blood pressure goes higher. That's part of what happens when you get older and the hypertension that develops. It's because of the loss of elasticity in those arteries. Sorry, a little squirrel there, but I guess I have to stop because that was done 5 minutes ago. Lab, head up there, pull your cats out. I'll just quickly go through a couple of them, show you the blood vessels. Yeah, there's not much to dissect. You basically grab the kidney, plug it in the side, there it is. So it's pretty easy to find the last of the arteries and veins. Oh. Diane, what's happening? How you doing? I forget what you had. Were you sick? Were you working?
I got COVID.
Oh, good.
But, because I work as a resident in the hospital, I got my 2 weeks in the hospital, which was too much.
Gotcha. You missed the exam too, right?
Yeah.
Take the exam last time? Was it over endocrine or reproductive development?
I'm
All right.
ready.
What's that?
I'm ready.
All right, well, let's do it right now. Then I've got Autumn, you're taking it tomorrow, Thursday. If viewers want to come in early Thursday, let's see, I've got 2 more videos to post. They're really a pain to get up for so long, man. They don't load up my equipment very well. But I will post them. I think I've got the end of female reproductive system and all development on videos. I've got 1 posted. I got the second one on my thing. I'm going to try to load it over and get one on YouTube.
Okay. So you want me to do that?
Thursdays, coming at 10,
I will come in at
11:30.
My alarm did go off. I turned it off.
Well, you can go after lecture as well.
I don't want to do all that. I like, because it'll be more fun.
Come in about 9:30 then. That gives you an hour and a half before class on Thursday.
Hey, I did get up at 6 this morning. But, then I went back.
I have to get up at 6:40. I have weight classes.
I hate
When
that.
you dropped in school. That's really a pain. My wife is a school nurse. She has to leave too early, so she had dropped. So I get stuck with those monkeys every day.
Oh my gosh.
It's
Yeah, I think I got an upper respiratory infection now.